We will just wait a couple of moments just to let everyone. The webinar will start now. Welcome and thank you for taking the time to join us today for this exciting webinar being brought to you by Scintica. My name is Gabriel Escalante, and I will be your host for today's webinar. Our webinar today is entitled Modality Review, the Basics of IntraVital Fluorescence Microscopy. Today's webinar will be given by Dr. Sarah Rapich, Product Manager within the Imaging Division at Scintica. Sarah has received extensive molecular imaging training in Europe and Canada since 2008. She holds a Master's in Science in Molecular Imaging and a PhD in Medical Sciences from the University of Antwerp in Belgium. Her doctoral research focused on the evaluation of novel radial tracers as biomarkers of treatment response in a colorectal cancer model using nuclear imaging technologies. During her postdoctoral fellowship at the Princess Margaret Cancer Center in Toronto, she studied the complex tumor microenvironment of pancreatic cancer using IntraVital microscopy. During this webinar, Sarah will introduce the fundamentals of fluorescence microscopy, review some example images, and focus on this technique's IntraVital imaging applications. As some of you may know, Scintica is a distributor of IVIM's IntraVital microscopy platform systems here in North America. Today's webinar is the first in a 4-part series, which will be spread out over the next few months. Please keep an eye out for the emails in the coming weeks with additional information about the upcoming webinars. Before we begin, I'd like to briefly mention a few housekeeping rules. We anticipate that today's presentation will be just under an hour, so we may not have time to answer your questions live. However, please feel free to submit your questions in the Q&A dialog box throughout the presentation, and I will work with the team to get them answered. We'll create a transcript of the questions and we will distribute this document within the next week or so. Please also note that we're recording today's session, so if for some reason you lose the connection or can't hear everything clearly, you will have access to the video file to review later. Without further ado, I'm going to pass things over to Sarah.
Thank you for that introduction, and a big welcome to all of you attending today's webinar on the basics of IntraVital Fluorescence Microscopy. We'll start off with fundamentals of fluorescence imaging, after which I'll introduce you to IntraVital microscopy systems, advantages, models, and some quantification methods. And we'll close off with key research applications of the modality. First off, I will place fluorescence imaging in the realm of preclinical imaging modalities. There are plenty of imaging modalities to choose from in preclinical research, and some of which are shown in this chart and plotted against their spatial resolution and their ability to visualize structures and/or these structures' functionalities. When choosing an imaging modality, you have to ask yourself what it will take to answer your research question. Do you need to be able to visualize a cell, what's happening inside that cell, how that cell is interacting with other cells? You will choose an optical microscope. If you can answer a question by discerning organs or looking at whole body, you can choose, for example, to use CT or MRI in order to look at anatomical structures. If you need to have some more functional information of those structures, you can choose to work with bioluminescence imaging or PET. Some of these modalities will also be combinable to get maximal information for your research. Today, for IntraVital microscopy, of course, we will focus on the left side of the chart, where we can resolve very tiny structures into the micron size. And we can also look at their functionality and cell-cell interactions and dynamics, for which we need to have a good enough speed of imaging. If you're interested in some of the other imaging modalities, please have a look at the Scintica website, as a lot of information is available there, as are some specific webinars. The general rule of thumb for any form of imaging is that the maximum depth you can image at will be about 10 to 100 times the method's spatial resolution. As we've just said, microscopy has the highest spatial resolution at micron level, but it also means it will have the lowest penetration depth into the tissue. This is mainly due to what happens to light when it travels through a medium. It will not just travel straight through and transmit to the other side. It can also reflect at the surface, scatter as it travels through, refract and backscatter, get absorbed by particles in the medium, turn into heat, or get absorbed by fluorophores in the medium and turn into emitted fluorescent light. Many of these properties can be imaged, and dedicated microscopes have been developed to do just so. Bright field microscopes can image transmitted light, and reflected light microscopes reflected light. Scattered light can be imaged by Raman spectroscopy microscopes, and absorbed light by transient absorption microscopes. To image fluorescent light, confocal and two-photon microscopes are good choices. These properties, they also depend on the medium that the light travels through and on the wavelength of the light. And the properties that mostly influence penetration depth are scatter and absorption. This can be demonstrated in the medium that is of interest to us here, which is in vivo tissue, using a simple experiment with laser pointers of different colors. In the pictures here, a green laser pointer at a shorter wavelength is compared with a red laser pointer at a longer wavelength. As you can see, the green light neither penetrates deeply into the living finger, nor does it scatter. On the other hand, the red laser light penetrates almost right through the finger and transilluminates the whole width of the finger. It also backscatters onto the hand holding the laser pointer. The main molecules in tissue that are contributing to this absorption are hemoglobin and water. In the shorter wavelength range, hemoglobin in deoxygenated and oxygenated blood plays the biggest role limiting tissue penetration of light, which improves with larger wavelengths in visible and near infrared light range. However, above 1,000 nanometers, in the infrared invisible light range, water, which did not play a significant role before in light absorption, now becomes a major contributor. In essence, to optimize penetration depth, you need to identify the optimal imaging window in this light spectrum. This slide shows the tissue penetration of the different colors in the spectrum, ranging from ultraviolet to infrared light, and shows that hemoglobin absorbs light maximally in the blue, green, yellow wavelengths, while water absorbs light maximally in the infrared wavelengths, which means that the optical imaging window for in vivo imaging is for light in the orange-red wavelengths of the visible light spectrum and in the near-infrared wavelengths, which corresponds to wavelengths of 600 to 1,000 nanometers. This is important to know when selecting fluorophores in fluorescence imaging, since fluorophores have to be excited by distinct wavelengths and will emit at a distinct wavelength as well. The way that this excitation emission works is through the absorption of the light by the fluorophore. The incoming light or excitation wavelength needs to correspond to the right amount of energy in order to excite electrons in a specific fluorophore. These excited electrons will end up at a higher energy state as they absorb this light, and as they're trying to return back to their ground state, will lose some of that energy to heat. As a consequence, the light that these electrons emit upon return to ground state will be one of a lower energy and thus longer wavelength. Note in the light spectrum here that there's a right shift from blue to red between excitation and emission spectra. The fluorophores in the tissue can come from different sources. They can be genetically modified fluorescent proteins, ranging from blue all the way to far red. Or they can be organic dyes, such as FITC and TRITC, which are green and red dyes, typically used for vasculature imaging. Or Alexas, which are also a vast range of colors that are typically conjugated to antibodies for targeted imaging. They can also be inorganic dyes, such as quantum dots, or fluorescence can originate from endogenous species, such as elastin and collagen, and the metabolic enzymes NADH and FAD. In this case, you're basically collecting autofluorescence from these species after exciting them with a specific wavelength. The other sources are typically introduced externally. However, it is possible to introduce them in a more permanent way through viral transfection into your cell lines or into your mouse model as a whole, in which case your fluorophore is again endogenous. The advantage of fluorescence imaging using fluorophores is that you're basically imaging very specific processes and in a very sensitive way because of the targeting. And it allows you then to also perform live dynamic imaging and combine the different fluorophores into multiplex imaging. In order for multiplex imaging to be successful, though, you need to choose your fluorophores carefully to minimize spectral overlap. There are tools out there, such as the fpbase.org website, that can help you with such selection. On this website, you can put together your fluorophores of choice and essentially calculate how much of the excitation wavelength for one fluorophore will also excite another fluorophore in case they are too close to each other in the spectrum. I've put together here 3 fluorophores that are very often used together in intravital imaging. There's FITC, which as I said earlier is often used for vasculature imaging. That one's in the green spectrum. There's M-cherry, often used to transfect cancer cells with, in the red spectrum. And there's Alexa Fluor 647 in the far red spectrum, often used to conjugate with antibodies. So as you can see, when I put in my excitation wavelength for green, which is 488 nanometers, I will excite 77% of my FITC, but also 8% of M-cherry, which is totally acceptable. Moving on to red, when I excite M-cherry with 561 nanometer, I will excite 64% of M-cherry, and also again 8% of Alexa Fluor, acceptable. Alexa Fluor 647 is excited with the 633 nanometer laser, and at the same time, besides exciting Alexa 647 with 62%, it will also excite 1% of M-cherry, which is negligible. So this spectrum works. So you've now chosen your fluorophores that you know can be distinguished from one another, but how will you actually achieve separating and imaging them? That is where fluorescence microscopes come into play, and they exist of specific components to make that happen, which are a dichroic mirror and a set of filters. The first filter is used to select a distinct wavelength for excitation of one of your fluorophores in your sample, here blue light. The dichroic mirror reflects the light onto your sample, from which emission light will be transmitted back. This light, here green light, will now be able to pass through the mirror, and then will go through the second filter to select a specific emission wavelength coming from your excited fluorophore, blocking all other unwanted light. This filtered light will then reach the detector, where it is translated into an image. You can do this for each fluorophore and are left with an image containing all 3 fluorophores, together yet separated. 2 specialized fluorescence microscopes most commonly used in intravital imaging are the confocal and the 2-photon microscope. They distinguish themselves from basic fluorescence microscopes, such as the one on the previous slide, by focusing a laser beam onto the sample and scanning at that point in a point-by-point way rather than illuminating the whole sample at once, thus increasing spatial resolution drastically. In order to collect the emitted light from the illuminated scanned area, the confocal microscope makes use of a pinhole before the detector. This pinhole rejects all photons emitted from outside the focus, as well as those that come from the focus but are scattered on their way to the detector. This way, only unscattered photons coming from the focal plane are able to pass through the pinhole and reach the detector, contributing to the overall signal. This inefficiency demands a high laser power for imaging, though, which could create unwanted photo damage, such as photobleaching and phototoxicity, and limits imaging depth to a maximum of 100 microns. In contrast, a 2-photon microscope does not require a pinhole, since all the emitted photons contributing to the signal come only from the focal point of the excitation spot, regardless of how much they scatter on their path to the detector. Greater tissue penetration up to 500 microns is reached with 2-photon microscopy, and fewer excitation events are required to achieve the same signal due to the improved collection efficiency, limiting potential photo damage in this technique. To understand why the signal in 2-photon microscopy comes only from the focal point of the excitation spot, we need to have a look at the principles of 2-photon microscopy, which differ from single-photon microscopy. As I explained earlier, single-photon excitation requires the absorption of a high-energy photon to excite an electron of a fluorophore to an excited state. When this electron then relaxes to its ground state, reaching the It emits a photon of light at a different wavelength than the excitation photon. In two-photon microscopy, reaching the excited state of an electron is achieved by the simultaneous absorption of two photons. The likelihood for this to happen is basically only reachable in the focal point of the laser beam, which is why there is no out-of-focus fluorescence from scattered photons, eliminating the need for a pinhole in the two-photon microscope. And since two low-energy photons with a long wavelength are used to create the energy needed to excite the fluorophore, the penetration depth will be higher. Now let's take a moment to summarize what we've learned so far, which is that optical imaging has excellent resolution but poor penetration depth, which is linked to how light propagates through biological tissues, where it's absorbed by hemoglobin and water, affecting the penetration depth. This leaves us with an optical imaging window for in vivo optical imaging between 600 and 1000 nanometers. Here, fluorescence is the most commonly used optical technique because it's so specific and sensitive, and it allows for multiplex imaging, where you can simultaneously visualize up to 4 fluorophores when you choose them carefully to avoid spectral overlap. Compared to basic microscopy, IntraVital microscopy is a rather younger technique, with a first publication in the 1930s using luminescent light IntraVital microscopy. Over the next few decades, fluorescence microscopy using fluorophores became more and more developed, leading up to the development of the confocal microscope in the 1980s and the multiphoton microscope in the 1990s, at which point IntraVital microscopy became more pronounced in cancer research. Before that, applications were also ranged from looking at the anatomy of living organisms to performing static in vivo process imaging and longitudinal in vivo process imaging. And then more recently, studying the immune response in capillaries, and of course, tumor metastasis and pathophysiology, as well as treatment response. As you can see, over this time period, the number of applications using IntraVital microscopy has grown exponentially. By now you know that IntraVital fluorescence microscopy can provide you with molecular and functional information at high resolution and at cellular level, as compared to some other imaging techniques that operate at a more microscopic scale. Other selling points for IntraVital microscopy are the fact that it can be used in situ, which is basically in the place of origin, that it provides you with spatial information, that it can be acquired in real time, and it gives you images that are quantitative. You can collect these images over time longitudinally, and this way you can also reduce the number of animals that you need in your experiment, as every animal can serve as its own control. And on top of that, information collected with IntraVital microscopy can provide new insights in the processes of disease staging, development, and treatment development and response. There are a few animal models being used in IntraVital microscopy, such as the fruit fly, the zebrafish, the chick embryo, and most commonly, the rodent, typically the mouse. Imaging fluorescence in these models can be done either through endogenous fluorescence when working with transgenic animals, or through the injection of fluorescent agents, such as transduced cells, conjugated antibodies, and fluorescent dyes. Focusing on the mouse model, where is it that we can actually apply IntraVital microscopy? Well, pretty much any organ, system, or structure that you can access optically, you'll be able to image with IntraVital microscopy. Gaining access can be done non-invasively or invasively. There are a few structures that can be accessed non-invasively, such as the ear and the eye. Pretty much every other organ and structure you want to image with microscopy, you'll need to expose surgically to be able to access it with your objective lens. You can do this by removing a skin flap and imaging your organ or structure of interest right underneath, or by extracting your organ of interest and imaging it that way. These techniques are typically not meant for repeated imaging time points and are mostly performed at endpoint only. In order to perform longitudinal imaging, imaging windows have been created, which can be implanted surgically into the tissue of the mouse over your organ or structure of interest. A few very commonly used window chambers are displayed here, such as lung chamber, dorsal skin fold chamber, mammary window chamber, abdominal window chamber, cranial window chamber, a bone window chamber, and an ovarian window chamber. A lot of these window chambers have detailed protocols that you can find in literature and that can help you guide through the creation of your own intravital model. Use these guidelines to find important details regarding imaging stability over time, imaging tolerability by the animals, and optical imaging conditions such as choosing the right cover slip thickness, typically a number 1.5, and the right field of view to be able to visualize your structures well. Nevertheless, creating your own intravital model will take a fair amount of time, dedication, and skills. I will now play a video of the installation of the dorsal skin fold chamber, which is one of the most characterized window chambers out there, so that you can get an appreciation of the complexity of a surgery, this one being one of the easier surgeries. This imaging window is made out of stainless steel, but you could also 3D print it with plastic material. Sutures are used to surgically implant it and keep it into place and close off the skin so that there's no infection coming in from the outside. Cancer cells can be injected locally and allowed to grow underneath the glass cover slip, which is held in place with a snap ring so it cannot detach over time. Once installed, you can image your window chamber mouse multiple time points, visualizing here up to 3 fluorophores simultaneously, looking at vessels, cancer cells, drug treatments, red blood cells, and many more. It's important to point out, however, that all of these manipulations can cause a disruption of biology. I'm not just talking about the surgery, but also the antibiotics and anti-inflammatory agents that you may be giving to your animal before and or after surgery. The stress that goes with the surgery, but also with the prolonged imaging and with the repeated injections. There's also repeated laser exposure to your area of interest to keep in mind. And if using antibodies, there could be antibody binding effects as well. And all of this can lead to a changed microenvironment. It's therefore really important to also include positive and especially negative controls in your experiment so that you can exclude any kind of manipulation related changes in your biology that you could mistake for effect or lack of effect. In this study, for example, they looked at macrophages after surgery to exclude an immune reaction that could be confounding their results. So after about 72 hours, they saw macrophage infiltration decrease back to baseline levels, at which point they could start their experiments. Once you have established, optimized, and characterized your intravital imaging model, however, its applications are pretty much endless. You can apply it in stem cell biology, oncology, drug development, immunology, infection, and inflammation, allowing you to study cell-cell interaction, cell trafficking, cell microenvironment interaction, in vivo, and over time. Now that you're ready to acquire images, you'll have to think about which imaging settings you will be using that will determine your image parameters and therefore your quality of analysis. Common image parameters to keep into account are image contrast, your sensitivity, your image resolution, the speed at which you're acquiring images, and the depth at which you're looking. I won't go into detail for each and every one of this because how you set them will be different for the majority of the softwares. But keep in mind that in general, imaging is all about trade-offs. You will always have to balance these parameters against each other. So it's important that you figure out what is the most necessary parameter for you to be able to perform successful analysis of your images. Overall, your goal should be to obtain an image with a good enough contrast for analysis, without any saturated pixels, with minimal bleaching, and with overall settings that you can reuse over different imaging time points and between different imaging subjects so that you can objectively compare the images that you've acquired against each other. There are a few conditions that can complicate your image acquisition and therefore analysis. You can't necessarily correct for and account for during image acquisition. You can take care of it during pre-processing though, which is the stage between image acquisition and image quantification. For example, motion artifacts is one of such parameters that are popping up often in IntraVital microscopy because you're performing live imaging. So because of the breeding of the animal, there will be a certain motion to your image, depending on where it is that you are trying to acquire this image. This, for example, is the lung, which you can imagine has a lot of motion artifacts. If you were to take an image without any correction for this motion, you'd end up with quite a blurry image with poor contrast, and your image analysis of these structures will be hampered. So there are certain ways for you to be able to correct for this motion artifact using specific software features and be able to end up with a crisp image for analysis. In pre-processing, you can also correct for spectral crosstalk. Even though you've carefully selected your fluorophores and you know that they're distinguishable from one another, there can be still some bleed through happening from one channel into another, such as here with the blue channel bleeding through into the green channel, showing nuclei where they shouldn't be. By using software that assigns a specific spectral signature to each of the pixels, you can group pixels with the same spectral signature together and subtract them from images where they don't belong, leading to an unmixed image such as below, where the blue has been subtracted from the green and no nuclei appear anymore in this channel, leading to a better separation of the fluorophores. Also, the presence of autofluorescence can diminish the contrast in your image and complicate quantification. So again, with some software applications similar to spectral unmixing from the previous slides, you can assign a specific spectral signature to your what you know is autofluorescence and not specific signal, and subtract it from your image to get a better contrast. So now that you have your acquired and pre-processed images, you can go and quantify these using commercially available softwares such as Imaris or freeware such as Image and Fiji, or software that comes with the microscope. The quantification that you can perform can be done either in two-dimensional images or in three-dimensional images if you acquired them as such. The analysis that you can perform ranges from cell motion tracking, where you can look at speed, acceleration, and track length for single cells. You can also quantify surfaces such as cell area, volume. You can also count these cells and quantify a cell number. You can also look at the distance between cells or distance to surface. That will help with quantifications in terms of cellular interactions and intracellular changes. You can also quantify vessels in your images, looking at leakage from these vessels, their density, number of branches that they have, their length and volume. And you can also see and quantify cells intravasating and extravasating from them or from lymph nodes or other structures. So depending on your needs, depending on the type of images you've acquired and the type of software you have access to, there really are a multitude of options for quantification and giving you the information that you need to be able to conclude something about your research question. So for the second part of this webinar, we can conclude that in order to perform successful intravital imaging, you need to choose the right system for your setup. You need to choose the right animal model and optimize it, and make sure that your window chamber quality is optimal for in vivo longitudinal imaging. As you're acquiring these images, you need to make sure that you position your animal well in respect to your objective lens. You need to make sure that your animal has a good breathing rate to reduce motion artifacts as much as possible. And you need to have thought about which imaging parameters make sense for your image acquisition, and that you can apply this over time and between animals for objective comparison. And then after you've acquired your images, you need to consider applying pre-processing corrections and which post-processing quantification is needed for you to be able to answer your research question. I'll now be showing some confocal microscopy intravital images of some key research applications. In general, intravital microscopy is most often applied in oncology and immunology research, but also often in order to unravel stem cell biology and characterize drug development. There's also really good mouse models out there of inflammation and infection, allowing for intravital microscopy research of that. And using more often the zebrafish and fruit fly models, developmental biology and regeneration are being studied with intravital microscopy. And then more recently, there's more research being done on tissue engineering and microbiology with the method. We'll start with some examples in oncology. The dorsal skin fold chamber is one of the best characterized chambers out there and has been used extensively in oncological research. You can compare it a little bit to a subcutaneous tumor model, as it's not quite in situ growth of cancer cells unless you're studying skin cancer, but it has allowed for a lot of interesting research and progress in terms of oncological development and treatment response. So here you see an example of a dorsal skin fold with a nice vessel structure through it. When you take an image with the confocal microscope after having injected some dextran to light up the vessels, and after having injected some cancer cells and allowed some time to grow, you can visualize these as red and green respectively. If you want to zoom in a little bit more into this area, you can distinguish microcapillaries as well and start to see more individual cell structures. So in this model specifically, we're looking at human non-small cell lung carcinoma. And what this system will allow us to do is to basically track changes in tumor growth over time or changes in vessel structure over time. So this is shown here. Images taken at day 7, 10, and 13 after a treatment regimen that instead of targeting the cancer cells is targeting the newly formed vessels that are supplying supplying the cancer cells with nutrients. So after applying this anti-angiogenic treatment and identifying your field of view that you want to track over time based on a vessel structure that is easily recognizable at each imaging time point, you can basically go ahead and track changes in green and red, being changes in the number of cancer cells or area or volume of cancer cells, and also changes in vessel morphology and dilation. Using the same window chamber model, you can also evaluate the treatment that targets the cancer cells directly, such as one delivered in nanoparticles. Even though they are very small, they can be visualized with confocal microscopy. Here, they're labeled with a red fluorophore and are shown against green labeled human triple negative breast cancer cells and far red labeled anti-CD31 antibody. This antibody labels the endothelial cells of the blood vessels and is here given the pseudo color of blue. That's not the light that is being emitted from the fluorophore. So here is evaluated how the nanoparticles, after injection into the bloodstream, reach the tumor and extravasate from the blood vessel to infiltrate the tumor site and start treatment. Multiple time points are collected, but here within the first 24 hours after injection. You can not very well appreciate how many red nanoparticles are present and where they're located because of the green cancer cells that are larger and brighter, as well as the nanoparticles. So in order to visually appreciate where the nanoparticles are at and how many there are, we can simply remove the green channel from the other ones and only look at the red and the far red channel. That way you can appreciate that indeed there are more nanoparticles entering and extravasating from the bloodstream and accessing the tumor site. Of course, when you're analyzing this image, you would analyze every channel separately. So this is just a tip for visualization purposes. Another commonly used window chamber model to study disease and drug development is the abdominal window chamber model. Typically, this is a circular chamber that will be sutured into the skin of the mouse over the abdominal structures of your interest. Here, you can see the pancreas, the spleen, and the kidney all in your field of view, which will typically be about 8 millimeters. Here are a few example images that come from this window chamber, showing in the kidney and in the pancreas, islet beta cells surrounded by their native blood vessel structures. And in the spleen, using a different mouse model, we can see the movement of monocytes and dendritic cells through and around the vessel structures. Both of these mouse models are transgenic mice, which means that they have been genetically engineered to express a certain color for a certain cell type. You can also perform gastric imaging with this type of window chamber and access small and large intestine. For example, here we're looking at colon cancer implanted into the cecum in green and vessels in red. So as you magnify closer into the tumor, you see that it's made up out of individual cells, but it's like a tight-knit community sitting in a nest of vessels that are providing nutrients to it in order to survive and grow. Removing the green channel, you can see just the vessels here. You can appreciate how unstructured they are. They're poorly formed, probably leaky vessels that are that were just quickly formed in order to supply the tumor of nutrients. Another organ worth investigating in the abdominal region is the liver. In this work, the pathophysiological drivers of non-alcoholic fatty liver disease were studied. This disease is chronic and affects people worldwide. However, its mechanistical drivers have not really been understood yet, so therefore effective treatment strategy has not yet been established. In this work, researchers developed a novel fluorescent lipid droplet labeling dye, the Fluor44 or SF44, in order to visualize individual lipid droplets. They combined this with an anti-CD31 far red antibody to visualize vasculature, and were able simultaneously and were able to establish a spatial distribution of hepatic lipid droplets in a mouse model that was put on a high fat diet to recreate the chronic fatty liver disease. So by performing repeated intravital imaging in this model, as it was on this high-fat diet, the researchers were able to showcase that these yellow hepatic lipid droplets over time on this diet were growing in size. And compared to the normal diet, they were definitely more prominent. There's also an abundance of immunology research studies using IntraVital microscopy. In order to study an immunological response, researchers here used the ear window chamber model. In fact, there's no window needed here, nor surgical manipulation, so it's one of the easier models to employ. The skin is just thin enough to be able to image right through with confocal microscopy. The research has used a lysosome M GFP model to study their immunological response in, because this model expresses a GFP in the lysosome M enzyme, which is primarily excreted by neutrophils and macrophages. Here on the right in this video, you can basically see the green cells move, which are the neutrophils and the macrophages, and you can see their location against the vessel structure, labeled with a anti-CD31 antibody conjugated with a far red fluorophore. So this mouse window chamber model was then used by the researchers to study the immunological response to the CARS protein, which has previously been shown to be secreted from cancer cells, where it then induces inflammation. In this study, the CARS protein was labeled with a far red fluorophore and injected intradermally into the ear. And then a time-lapse video was made over the next 6 hours, imaging the same region and tracking the recruitment of neutrophils and macrophages to the site of injury. As you can see, over time, more and more immune cells are being recruited, and they're populating the site of injury. Other popular immune cells to study are, of course, the T cells and the B cells. And without going into too much detail about the background of this study, I wanted to show you this video where you can appreciate T and B cells extravasating and intravasating from this lymph node in the hind leg, and allowing you to, over time, basically track their movements and population. Another application is in stem cell biology. For example, bone marrow transplantations, which are widely used in patients with blood disorders and cancers, can be studied using intravital imaging. You can do so in the bone marrow of long bones, such as the femur, for which there are also window chambers. But more commonly, the calvareal bone in the skull is used because it's easier to access. And there's plenty of bone marrow to be studied there as well. So as you can see here, the skull is exposed, and a cover glass is placed over it that allows for optical imaging of the calvarial bone, and you can nicely see all of the large veins and the sutures running through this. On the right are some longitudinal wide area confocal images of this same bone. You can kind of recognize the structures up here. And what is imaged is our bone marrow cells transplanted from one mouse into the other and tracked over 4 days since transplantation. So the researchers were able to see an increase in transplanted bone marrow cells from day 1 after transplantation up to day 4 after transplantation, where cells were not only increasing in number, but they were also distributing more widely across the bone marrow, and they were forming clusters that were also increasing over time. They also collected a video to observe proliferation events during a 5-hour time lapse. So arrows point towards transplanted bone marrow cells that are undergoing proliferation, which of course contributes to this increase in the number of cells and clusters that can be seen over time after transplantation. Studying infection is also important, especially in this day and age of COVID-19. A good place to study such an infection would be in the lung. Now, a lung chamber is not your typical window imaging chamber. It requires a bit more of an advanced setup because you can't just place the chamber over an exposed lung. It would deflate and your mouse would stop breathing. So you'll need to intubate your mouse before you can do imaging. And as you're performing imaging, as you can see in this video on the right, you'll need to basically suck the lung up against the glass so that it's stabilized and inflated. And this will not only help with the imaging, but also with the mouse breathing. For example, by imaging red labeled neutrophils in the pulmonary microcirculation, you can evaluate the level of sepsis in the lung. You could also interest yourself in tracking red blood cells in the lung microcirculation. And in post-processing, you can then analyze the path of these red blood cells and their track length, as well as their speed, with red being faster moving to green being slower moving red blood cells. And the last application I'll discuss will be studying inflammation. Here, the cranial window chamber model was used to study neuroinflammation, which is often implicated in neurodegenerative diseases such as Alzheimer's disease and linked to blood-brain barrier dysfunction. In this study, researchers used the lysosome MGFP mouse that I described earlier that allows for the tracking of neutrophils and macrophages as they express GFP in order to assess the level of neuroinflammation in a control mouse compared to a mouse where they induce neuroinflammation by intracranially injecting an acid. They then performed longitudinal intravital brain imaging of this mouse model, capturing images at day 1, 4, 7, and 10 after induction of inflammation. When comparing these images, they could see that the level of green cells, therefore the level of inflammation, was higher in cells in the mice treated with acid than it was in the sham mice. Also, interestingly, they could observe that the distribution of these cells changed over time, whereas the number of cells remained rather constant, which meant that the neuroinflammation was persistent over time. Here are the same regions, but then at a higher magnification, so you can better see the individual cells and appreciate that the number of cells is very different between untreated animals here and animals treated with acid, therefore having neuroinflammation here. To identify changes in vascular permeability, which would be meaning that there is a blood-brain barrier dysfunction. Longitudinal imaging was extended until 30 days after neuroinflammation. And by using an injectable dye that stays in intact blood vessels but is able to leak out of compromised vessels, researchers were able to show that where at day 7 all the vessels were intact, come day 14, dye was starting to leak out of the vessels, and leakage was therefore observed. The leakage was still present at day 21, and by day 30, it was accompanied by an influx of the lysosome M positive cells that were there attracted to the inflammatory response caused by the blood-brain barrier disruption. I just wanted to end by pointing out that sometimes using only one imaging technique will not be enough to answer your research question. So you may want to combine a few preclinical imaging modalities in order to perform multimodal imaging. For example, if you need to reach a deeper depth than what you're able to reach with confocal orthophoto microscopy, you could perform optical coherence tomography as an added imaging modality to give you complementary information about depth. You do have to be careful when choosing which two modalities you want to combine regarding your window chamber, because if you're going to work with stainless steel window chamber, it's going to be hard for you to perform CT or MRI imaging. Just something to keep in mind. And overall, it will always be a good idea for you to correlate your obtained results in vivo with ex vivo stains and validate the results that you have obtained and quantified with IntraVital imaging. I hope I've been able to show you that IntraVital microscopy, even though based on old technology, has come a really long way in the past decades, especially with the introduction of the confocal and the two-photon microscope. I hope you've also firsthand witnessed how it can be applied in a multitude of different research fields and how it can be of assistance in uncovering very dynamic processes that can answer very complex research questions. We've reached the end of this webinar, which is the first one in a series of 4 focusing on IntraVital microscopy. If you would like to learn more, please do sign up for the other webinars if you have not yet, because we have much more materials that we still want to share with you. And meanwhile, if you have any questions, then please do not hesitate to reach out to us on the email address or website shown on this slide. Thank you.
Thank you, Sarah, for the great discussion today. We would like to ask a couple of quick poll questions here, learn more about you and the work that you're doing. Okay, so the first question is going to be, are you currently using IntraVital microscopy in your work, or have you in the past? And the second one will be, are you using any other types of imaging in your research? And you can choose all that apply. We'll take a few moments for everyone to answer the poll. And for the meantime, I just want to remind everyone, please feel free to ask any question in the Q&A dialog box, and we will create a transcript in the future. Thank you. Okay, I will end up the poll session. So we've reached under an hour, a mark for our session today. And to be respectful of everyone's time, we're going to wrap things up. As mentioned at the start of our webinar, we'll be sure to answer any type of questions in the written transcripts, and we will work to to get this out to you over the next week or so. I'd like to thank again Sarah for the wonderful presentation today. I trust that we have been able to provide you with some relevant information about the basics of intravital fluorescence microscopy. As I mentioned at the beginning, today's webinar was the first in what will be a 4-part series. Please keep an eye out for emails in the following week for the next portion of the series. If in the days and weeks to come, you have further questions about the modalities discussed today, I encourage you to reach out to us here at Scintica. We will be happy to discuss further. We will welcome the opportunity to discuss your specific research goals and how any of our imaging systems could help move your work forward. Thanks again to all of you for taking time out of your day to attend our session. We look forward to seeing you at a future Scintica event. Have a great rest of your day.